Today we will be demonstrating hanging drop technique in forming embryoid bodies. Wipe down the hood and any equipment used with 70% ethanol. Always make sure to use proper aseptic technique. Begin by using a sterile pipette to transfer 17 milliliters of PBS or phosphate buffered saline into a petri dish. Switching to a new sterile pipette, transfer 2.5 milliliters of DMEM containing embryonic cells to a separate sterile petri dish. Set a micropipetter to 27 microliters. Using a sterile pipette tip, place each 27 microliter drop on the underside of the lid of a second petri dish. Place each drop by touching the tip of the micropipetter to the surface of the underside of the lid. Dispense each drop by holding the micropipetter vertically to avoid dragging. Continue placing drops until the entire surface is covered, making sure the drops are evenly spaced and far enough away from the edge. Check to make sure the PVS covers the entire bottom of the dish. When the surface of the lid is covered with drops as shown, flip over the lid. Be careful not to disturb any drops, bringing the petri dish together to form a hydration chamber.
One week later.
After the appropriate amount of time, remove the petri dish from the incubator and place into a sterile hood. Remember to wipe down the hood and any equipment necessary with 70% ethanol. Assuring proper aseptic technique, remove the lid in one smooth motion. Using 1 milliliter of DMEM, begin to wash the drops down from the lid into the rim. Continue washing until all embryonics are washed down into the rim. Transfer all media containing embryonics into a centrifuge tube. Add new DMEM until a final volume of 3.5 milliliters is reached. Transfer the entire contents of the centrifuge tube into a 25 centimeter squared culture flask. Place the flask into a 37-degree incubator.
Day 2.